Certainly she's been prolific author of many papers regarding many different subjects. First knew Michele when she was at Disney World. And she's been 2 or 3 places since then. Michele.
Thank you. Thank you, Dennis, and thank you to the IEF again for giving us the opportunity to have a lot of our African colleagues here interacting with the international group, and I think some great ideas have come out. So today I want to follow up on David's excellent talk about MTB complex infection in our case, in free-ranging African elephants in South Africa, and what we're trying to do to develop some novel diagnostic approaches. Oops. Just acknowledgement to my group, and of course the many different groups that help support our work, never done in isolation. This probably is, the numbers are a bit old now, but we know that MTB infection in captive elephants occurs worldwide. And most of the cases that have been described in the literature have been in Asian elephants. We have had 2 cases of human tuberculosis in African elephants here in South Africa at the National Zoological Gardens, and that has been typed, as David said, to some of the predominant local strains. So it's occurring in zoos and in captive or managed elephants around the world. So it happens obviously in both species. But the question's always been, what about TB in wild elephants? We know how difficult that can be to sample them and screen them and really follow up. So it was, I believe, back in about the early 2000, maybe 13, there was a case of a MTBC infected death in an African elephant in Kenya that had been an orphan and had been hand-raised. Unfortunately, they did not speciate it, so we don't know if it was MTB or M. bovis, but the speculation was that it had occurred very similar to the situation with mahouts through some of the handlers and close contact of that animal. Since then, there have been increasing numbers of sporadic cases in wild Asian elephants found in national parks, both in India and Sri Lanka, and I know many of our colleagues here can go into that in much more detail. So it does happen, but then the question is, how often is it happening, and is it really a concern? So that brings us to South Africa. Other than TB in elephants in our zoos, we really had not diagnosed TB, either M. bovis or MTB, in any of our wild animals in South Africa. But in 2016, you can see on the top there, for those of you, obviously we're in South Africa, we're kind of just above Pretoria, but there's Kruger Park up in the northeast corner. It's a very long, skinny park. And this elephant was found dead about a third of the way up the park, just kind of out in the open. As I had mentioned to some of our colleagues, it's very rare for us to find dead elephants. So we investigated it. As you can see, it was very thin at the time. We didn't find any wounds. Sometimes they die either from gunshot wounds on rare occasions or fighting with other bulls. There was no evidence of any kind of trauma. So then, of course, we started to investigate. And when we did a field necropsy, and when we opened this animal up, it became quickly apparent that it had generalized tuberculosis. So most of its lung was gone. We had miliary granulomas, and in some areas it became confluent. And obviously the cause of death looked to be associated with that. And it had disseminated to other organs as well. So this was the first time we ever had a case that we actually suspected of tuberculosis in Kruger. For those of you that are not aware, Kruger is endemic for Mycobacterium bovis. So we have more than 15 different wild species that are infected with M. bovis. So we got quite excited because we thought maybe this was a case of M. bovis. But as we did additional diagnostics, we did some serodiagnostics, direct PCR and culture, and over and over again, it confirmed that it was M. tuberculosis, so the human form of tuberculosis, which surprised us a bit considering where we found this animal. So that, of course, prompted the question of, was this a rare single occurrence, or is that something that we just haven't been picking up in our wild elephants? So for the last few years, we've been doing an increased surveillance in our Kruger elephants, and that includes, again, opportunistic necropsies whenever we do have an elephant that's either been shot or dies that we can get to, and we have found one incidental case of M. bovis in an animal. Again, that was not its cause of death. We don't know how it would have progressed, but we did culture it out of tracheobronchial lymph nodes. We've done a retrospective serological survey using the ChemBio DPP, and we get about a 6% seroprevalence. Again, with the confidence intervals going up to about 12%. So that's a bit surprising. Again, seroprevalence doesn't tell us if it's M. bovis or MTB. And then we've taken on active surveillance, which again is a bit of a slow process, but I think we're up to about 35 animals now that we've done so far in this program, which means actually immobilizing, and we're targeting bull elephants because we believe that, again, both based on the seroprevalence and the cases we've found, that it's more likely to appear in our bull elephants. So we immobilize the elephants. We have a field unit, as you can see here, with a 3-meter flexible endoscope, light source, screen, everything set up. We intubate our animals, and then we actually perform bronchial alveolar lavage. We use that for cytology, for culture, as well as then doing a trunk wash, which is just simply a manual procedure, again, aspirating out the fluid. And then, of course, we're taking blood samples to try and correlate all of these results. What I want to share with you is some of the things that we're starting to develop. We still have a long way to go, but when we collect these samples, for instance, a trunk wash and the lavage, and on occasions tissue, we now have at our facility in Kruger the Cepheid Gene Expert, which is a common benchtop PCR that's used for human TB diagnosis. And it's literally not much bigger than a laptop. You can get these cartridges that are basically a PCR in a cartridge. You take your trunk wash or your lavage or tissue homogenate, put it in a buffer, and then you put it, in the human case, it would be sputum. You put it into the cartridge, and you can actually have a PCR result in less than 2 hours, including some of the antibiotic resistance genes. So that has been very, very useful. I'll show you what kind of preliminary results we have with that. And it's something that we think, certainly in Africa, because of the number of TB clinics around African countries and in rural areas, might be a tool that wildlife veterinarians can use. We're also doing mycobacterial cultures. We've been working with a group in the UK that now has a new liquid medium supplement that can enhance the growth of mycobacteria, and the time to positivity in culture is about half of what we normally have, which is normally about 6 to 8 weeks. And then, of course, we use molecular techniques to speciate. So we're quite excited about this work. What we've done is we've taken uninfected elephants, collected trunk washes and bronchial alveolar lavage samples, and then we've been spiking it with known amounts of both M. bovis and MTB as a proof of concept for using things like the Cepheid Gene Expert, because it's simple enough, even I can do it. So any one of us in the room with 10 minutes of training can do it. And what we found, this is, again, looking at specific genes that are found in M. bovis and MTB, okay, is that even as low as 2 colony-forming units per mil of MTB in a sample, whether that's sputum, trunk wash, or whatever, can be detected. This is just the number of kind of the threshold for replication in a PCR. Many of you know about CT values. If you don't have amplifications, up around 30, 35. So you can see that as the number of bacteria present go up, you can get very rapid and very clear results. Based on the test that's looking at these particular genes, there's fewer of those genes in M. bovis, so our lower limit of detection is 30 bacilli in a 1 ml sample. So again, it can be very, very sensitive. One of the disadvantages of this is it does not distinguish between MTB and M. bovis, but it can tell you if the DNA is present, and then of course your index of suspicion goes up. Like I said, you literally can go from your elephant to your gene expert and have those answers very quickly. So we're very excited about that. Of course, it's important epidemiologically, as David said, to know, is it M. bovis or MTB? And the nice thing about those cartridges is that you can actually extract DNA and then do additional genotyping or speciation. So most of the time we do use either spaligotype, and more people are using whole genome sequencing from an epidemiological standpoint. So going back to our bull in Kruger, we did do whole genome sequencing, and the nice thing about that is, again, it helps you understand a little bit about the potential sources. And as David mentioned, again, this elephant, the isolation was clustered with the human strains that we commonly see in the provinces of Limpopo and Mpumalanga, which is where Kruger is. So I think it's really something important that helps us understand, especially in chronic diseases where we're not going to be able to trace the source because those animals have been infected for years. And then the other thing that we're developing is, again, in humans, they do a lot of cytokine work, gamma interferon. So we're trying to adapt some of those techniques to field techniques that can be used for elephants. So we use the human quantiferon tubes for stimulation. It's like doing a skin test in a test tube, basically, with whole blood. And then what we've been looking at is how can we then use that blood sample with either cytokine protein ELISAs or gene expression to detect the host response. Again, an animal that's never been infected or exposed to these organisms shouldn't have a response, just like in a skin test. But those that do, we should be able to pick up immune responses. So again, an exciting area of opportunity to improve our diagnostics. And again, it's easier sometimes to do, well, it's definitely easier to get blood than do a bronchoalveolar lavage, especially in the field. So I think one of the things that we need to think about is how do we continue to try and understand tuberculosis in elephants, and especially in free-ranging populations. So this is a map of areas around the world that have very high human prevalence of TB. And of the 30 high TB burden countries, 22 are in elephant range countries. So like David was saying, the threat of humans to animals, oftentimes when we're talking about zoonotic or emerging diseases, we're thinking about what animals are doing to human populations. In fact, we should be thinking the other way around. What is the human population threat? How do they threaten the animal population? So South Africa has an extremely high TB burden. It's estimated that about 80% of the population is infected, although most of those cases are latent. So we talk about, again, some of the captive elephants and things in southern Africa. The threat is going to be from people rather than the elephants presenting a threat to people. So I think TB is one of these things where it's a tip of an iceberg. It certainly is a huge concern for global human health, and what we need to really think about is, again, integrating animal health with human health. For the first time, WHO in 2017 came out and said, if we can't control M. bovis and other Mycobacterium in animals, we're never going to be able to eradicate TB in people. So hopefully the human community is kind of catching up with the conservation veterinary community in looking at using One Health principles. And that's it. Thank you.
Thanks, Michelle.